today what I'm going to talk about is a little bit about geobiology. And so this idea, this concept of geobiology has to do with how basically it's the study of how biology influences geologic processes and vice versa. So the interaction between the geosphere and the biosphere. And there's a kind of a subset of geobiology that is termed geomicrobiology that specifically refers to the interactions between microorganisms and rocks and minerals broadly, or chemistry, the chemistry of the planet. And here you can see up here a couple of kind of nice little illustrations of that. These are both rock samples that I've collected from using deep sea submersibles. The one on the left here is barium sulfate. It was collected from the Gulf of Mexico. And you can see here all these little filamentous structures here. These are sulfur oxidizing microbes, Beggiatoa, that are basically encrusted in a barium sulfate mineral deposit here. And we think that the microbes were important for actually kind of catalyzing the precipitation of at least some portion of this deposit. And over here, so over on the left side, we think microbes are important for forming minerals. Over here on the right side, these are actually, this material here is this kind of tan-colored stuff. That's calcium carbonate that is actually formed at methane seeps. And we think that there are various microorganisms that are involved in the anaerobic oxidation of methane that change the chemistry such that it causes these rocks to form, these carbonates to form. And then later, these rocks can actually be colonized by a different group of bacteria that produce sulfuric acid as part of their metabolism. And we think in that case that they actually cause weathering on the ocean floor of these rocks, and it leads to all of this pitting and dissolution and so on that you see actually occurring on the sea floor. So some microbes are involved in making rocks, and some microbes are involved in dissolving them. But today, so those two are two examples of carbonate rocks and rock known as barite. But today I want to talk a little bit about microbial involvement in these rocks that are called phosphorites and its association with this metabolism called polyphosphate. And it's a type of metabolism that's used by lots of organisms, but also in particular, we think that these giant sulfur oxidizing bacteria are important. So I'm going to kind of first introduce these concepts, give a little bit of background about these three seemingly unrelated things, and try to convince you that they're all sort of related. And then I'm going to talk about kind of phosphorites and bacteria in deep time, so going back into the deep geological record. And finally, I'm going to tie this into the kind of the theme of this presentation and talk a little bit about some work that we've done looking at polyphosphate in the deep ocean. So phosphorus is a limiting nutrient for primary production, and it's very important for biology because it's a component of DNA. It's necessary for the synthesis of ATP, phospholipids, and a variety of other different biological molecules. You can see kind of a graphic illustration of that down here in this lower right-hand panel. This is a lake that's been experimentally separated in two, a lake in Canada. And up here on this side, you can see this is kind of the dark normal color of the lake. And over here, this is this kind of pea soup looking color here. It's basically a bloom of algae and cyanobacteria. And the difference is that only nitrogen has been added to this side of the lake, and nitrogen and phosphorus has been added to this side of the lake. And so you can see this kind of really nice illustration of the influence, the role of phosphorus there. I think my battery is dying a little. Do you have another laser pointer? No. Okay, well, hopefully you can see this one. Okay, and one of the reasons that phosphorus is also a limiting nutrient is that it really basically enters the hydrosphere and the biosphere through the relatively slow weathering of phosphorus-rich rocks. So unlike with nitrogen, where there's a biological mechanism for taking up nitrogen from the atmosphere, with phosphorus, these rocks basically have to weather and dissolve more or less to get the phosphorus out of some small component of the minerals here that contain phosphorus. And once that phosphorus enters the hydrosphere, this is just kind of a schematic that shows at least some of the major parts of phosphorus cycling, certainly not all of them, but basically that phosphorus from the weathering enters some kind of dissolved phosphate pool in rivers and then transported to the ocean, where biology takes up the phosphorus, and then when that biomass dies, it's quickly returned to phosphate, recycled, and taken up by other organisms, and because organisms need phosphorus, it's very efficiently taken up by the biosphere. But there is some component of it that can, phosphorus that gets absorbed onto iron oxide minerals, so various different clays and rock material can have phosphorus absorbed to it. And in some cases, that phosphorus can actually be buried as it's absorbed to those mineral grains. But fortunately for the biosphere, in most cases, or in many cases, once those iron oxides get buried to a certain depth, microbes basically reduce the iron oxides and dissolve the iron, and that releases the phosphorus back into the water column. And so that's called the iron redox pump, is what it's termed. And so that's a mechanism that returns phosphorus back to this dissolved phosphate pool. There is a little bit of kind of background production of calcium phosphate minerals broadly in ocean sediments. And so you get a little bit of phosphorus that gets buried as calcium phosphate or as other different types of buried phosphorus. And so I term that here background burial. But the thing I want to talk about today are these kind of interesting and enigmatic deposits that are called phosphorites. And so phosphorites are basically deposits that are really rich in phosphorus, mineral deposits. And we don't really understand how these phosphorites form. But we do think that they're an important part of phosphorus cycling, and I'll tell you in a second why they're kind of relevant to us. And I have this picture here that shows a bunch of different fruits and vegetables. It's just kind of illustrating that we think there are lots of hypotheses out there about how these phosphorites form. I have a couple of favorite ideas about it, but there are lots of other, mechanisms that have been proposed, and I'm just going to talk about kind of one subset of those today. So what are these phosphorites? These are basically rocks that contain greater than 6% P2O5, whereas most kind of marine sediments and marine sedimentary rocks contain less than 0.3 weight percent P2O5. And you can see some pictures here of these kind of grains and nodules and crusts and things that are basically made up of so-called apatite group minerals that contain calcium and phosphate, but also some other stuff too, like carbonate and fluorine, oxygen, so on. There's lots of substitutions that can, elemental substitutions that happen with these apatite group minerals. But another apatite mineral is actually our bones, right? So the calcium phosphate in our bones, those are hydroxyapatite with other biological components to it. But basically that's a hydroxyapatite mineral, our bones. So these are similar in composition and mineralogy to our bones. These deposits are often so-called reworked and concentrated by various different types of sedimentary processes. But how the actual phosphatic mineral forms in the first place is still something that's not well understood. And this term phosphogenesis, it basically refers to a process or processes that result in the kind of the precipitation and accumulation of these mineral deposits. And so, but who cares? Why should we care about these phosphorites? Well, really, these phosphorites, once they're uplifted on land over millions of years, they're basically a source of phosphorus for us, and they're in fact the primary source of phosphorus for all of our agriculture that feeds all the billions of people on this planet. And you can see here in this graph here, over time, we have an increasing reliance on phosphate rock as a source of phosphorus for agriculture. And there are lots of uncertainties about it, but it's thought that by 2030 something, that there could be peak production of calcium phosphate minerals. By production, I mean basically mining production. So as these kind of high quality phosphate reserves get mined out, then production will decrease and prices will increase and so on. And we could face some kind of phosphorus crisis in our lifetime or our children's lifetimes. We most likely will, in fact. So this is kind of a non-renewable, at least right now, resource. So it's something that we should be paying attention to, and something we still don't really understand in terms of how it forms. Okay, so now I'm going to shift a little bit and tell you about this, this so-called metabolism of this molecule called polyphosphate. And then I'll bring it back to the phosphorites and tell you how it's related to them. So polyphosphate is basically this polymer of phosphate groups that's linked by these so-called phosphoanhydride bonds. And basically, in terms of its composition and its bonds, it's very kind of similar to ATP, right? So you have this chain of phosphates here in ATP where you have a lot of potential energy that can be linked to all sorts of biological processes, right? And so polyphosphate is this, it can be a huge chain of these phosphates that can also contain a lot of energy, and it can be stored in the cell for quite a while, as opposed to ATP, which is turned over quite quickly. And most organisms actually make polyphosphate, and oftentimes it's actually just produced as kind of in trace concentrations, or it doesn't stick around in the cell for very long. There's not very much of it in most cells, but some bacteria and some eukaryotic cells actually accumulate lots and lots of this polyphosphate. So it can be up to 20 weight% of the cell. And oftentimes this is a response to various different types of stress conditions. Those stress conditions can be a whole variety of things like salinity extremes or pH or who knows, maybe pressure is even important. But many of the organisms that are kind of well studied with respect to polyphosphate storage and accumulation, the ones that accumulate lots of it, actually do it in response to changing oxygen or kind of redox conditions. And this is showing a culture of some bacteria, and all these kind of bright yellow spots are actually globules or inclusions of this polyphosphate. I think I've run completely out of battery. One of the systems where this polyphosphate metabolism has actually been quite well studied is in these so-called phosphate removal reactors in wastewater treatment. And so in this case, you have an organism that's called Camelobacter phosphatus in the lower right-hand corner here. And then this organism, basically, it accumulates lots and lots of this polyphosphate under aerobic conditions. And then once it accumulates that polyphosphate, the reactor is kind of cycled to anaerobic conditions, and it breaks down that polyphosphate, uses the energy in order to take up a bunch of organic carbon and store it as these so-called polyhydroxyalkanoates. So this is like a big polymer of organic carbon. And then it uses those when it switches back to the aerobic phase. That gives it kind of a competitive advantage. So it uses the polyphosphate that it stores as kind of this battery or energy reserve in order to jumpstart its transition from aerobic to anaerobic metabolism. Is this one a laser pointer? This one? Don't know? Oh, good. Okay. So now, that last slide I was talking about, the polyphosphate accumulating bacteria in the wastewater system. In nature, there are also polyphosphate accumulating bacteria that we think are important for the geochemistry of the phosphorus cycle. And one of those is this organism here called Thiomargarita. And Thiomargarita is the largest of all known bacteria. Here you can see a fruit fly for scale, and these are individual Thiomargarita cells, so they can get up to about 1 millimeter in diameter for one cell. Right, so you can see these bacteria, one cell, with your naked eye, no microscope needed. And here, you can see all these little spheres in here. These are actually inclusions of elemental sulfur. And most of the volume here of this cell is actually a vacuole that contains nitrate. And these organisms, they use hydrogen sulfide as an electron donor in their metabolism. And they use oxygen or nitrate in order to oxidize that hydrogen sulfide for chemolithotrophic metabolism. So they can use oxygen, take that oxygen and oxidize the hydrogen sulfide, or they can use nitrate to oxidize the hydrogen sulfide and convert it either to N2 or ammonia, depending on the pathway that they use. And these Thiomargarita are closely related to Thioploca, which we find off Chile, and also Beggiatoa, which we also find here. So in addition to all of these elemental sulfur globules that are found here in the cytoplasm, also there are lots and lots of other storage inclusions that are present in Thiomargarita, including granules of polyphosphate. So you can see all these spheres here that are stained red. That's all inclusions of polyphosphate. And interestingly, there's a correlation between where you find accumulations of these sulfur bacteria and where you find modern phosphorites. So here, this map is showing some sites of where we think phosphogenesis is happening on the ocean floor today. Also, that's the red circles here. And then the open circles are basically relict phosphorites, so ones that were formed in the recent past here on the ocean floor. Here off of Chile and off of Namibia. We see also populations of giant sulfur bacteria in these sites. And so this kind of association was looked at by Heide Schultz and colleagues that basically they looked at they took some Thiomargarita into the lab, even though it's not in culture. They just took some cells, and they showed that under oxic conditions, basically you have phosphate taken up and stored as polyphosphate, and then under anoxic conditions, or perhaps sulfitic conditions, the polyphosphate is broken down and the phosphate is released to give you this kind of spike in phosphate. And then they took that to the environment and tried to basically use it as a mechanism to explain this association that you see between Thiomargarita. So this is basically, here this is depth in the sediment. This is a core from off of Namibia. This accumulation of Thiomargarita cells that correlates approximately with this kind of spike in pore water phosphate, and also this kind of spike here, this accumulation of calcium phosphate mineral. So this is apatite precipitates that are in the sediment, right? And so the idea is if you get a high enough concentration of phosphate, you get conditions that are supersaturated with respect to the apatite mineral, and that causes it to precipitate, right? And so the idea here is that Thiomargarita liberates its polyphosphate under some conditions, dumps a bunch of phosphate out into the pore waters in a kind of restricted zone that allows the concentrations to exceed a certain point that causes the mineral to precipitate. And some additional work by Goldhammer and colleagues showed basically also that they could use a label, a radio label, to show that basically this phosphorus, in comparison with sample that was killed, that a lot of this label ended up in the organic fraction in thiomargarita, in polyphosphate in thiomargarita, and also in the authigenic apatite, so the mineral that was forming in the sediment, as compared with the killed control here. So the idea again is that phosphate is being taken up by these bacteria, stored in the cell as polyphosphate, and then under certain conditions, it's basically used up, and this phosphate is released and concentrated here, and then that leads to the precipitation of the apatite. But there have been other papers out there, other reports that show, so off of Chile here, this paper from 2010 showed that thioploca, there are correlations between thioploca and this kind of increase in pore water phosphate that's kind of the same magnitude as what's seen off of Namibia. But here, even though there are phosphorites off of Chile, in this particular case, the phosphorus was mostly associated with iron and not with phosphatic minerals, at least at the particular sites that they studied. So here, it doesn't seem like the association is so clear, and additionally, they couldn't stain any polyphosphates in thioploca. So if polyphosphates have been found in thiomargarita and in Beggiatoa, but not thioploca, at least so far. So is thiomargarita the real kind of player here? Is it really the important one? Is just the kind of phosphate release from polyphosphate really important? There's still kind of a lot of open questions there. But we think that this polyphosphate metabolism is linked to this phosphogenesis process. Okay, so now I want to take you, now that you kind of have the background of this phosphorite polyphosphate story, I want to take you a little bit back into the geologic record and look at some kind of interesting features of the kind of the deep time record of these phosphorites and some of the fossils of microorganisms that are actually found in them. So before I do that, I want to remind you here about this oxygen aspect of the polyphosphate metabolism. So lots and lots of these bacteria that accumulate polyphosphate, they do it, they're basically selected for under conditions that have kind of very dynamic redox conditions, or so dynamically changing exposure to oxygen, or gradient conditions where you have basically potential electron donors in terms of reduced fluids that are kind of fluxing into an oxygenated environment. So here is in the deep ocean off of Costa Rica, there's basically sulfide that's kind of fluxing out of the deep here and into oxygenated waters. And you see the alvin here is basically kind of slurping up these mats of bacteria that are growing here, where the sulfide is kind of seeping out along fracture surfaces in this rock. And here, so this is at a depth of a couple thousand meters. Over here, this is off the banks of the Mississippi River near my office in Minnesota. And here you have anoxic groundwater that's coming out along the banks of the Mississippi, and where it encounters oxygen, you have all these iron oxidizing bacteria that make this kind of rust-colored deposit here. And these bacteria here, these iron oxidizers, also contain polyphosphate. And we don't know for sure that they use it in response to oxygen changes, but that's what's thought to be the case here as well. So these organisms that accumulate polyphosphate, oftentimes they do it in response to these kinds of dynamic conditions with respect to oxygen. And the idea with, in particular with thiomargarita, is that under oxygenated conditions, phosphate gets taken up and stored in the cell as polyphosphate, and then under anoxic conditions or sulfitic conditions, then that polyphosphate is broken down and the phosphate is released. And if you see also this figure at the bottom here is showing basically seasonally, over time, that you have these kind of oscillating conditions in terms of the oxygen that's available at the bottom waters where you have Thiomargarita. And that's really, I think, why it's actually so big is, you know, it's basically storing its electron acceptor and waiting for these kind of gradients to change around it because it's not motile, right? And so Thiomargarita and a lot of these bacteria, they're basically, you know, they're adapted to conditions that are very kind of dynamic with respect to oxygen. Okay, so that's in the modern. Now I want to shift your thinking a little bit into deep, deep time, okay? So what this is showing here is basically the origin of the Earth at 4.5 billion years would be right around here, and here's today. So these are each billion years, okay? And what this is showing is basically the percent of present-day oxygen over here. And here we start off in the Archean, so this is early Earth, right? We have basically no oxygen, very, very little oxygen, right? So we had an anoxic atmosphere. And then around about 2.5 billion years ago, there's this dramatic rise in oxygenation that's generally linked to the advent of oxygenic photosynthesis, or at least at least a time when the effects of that oxygen are allowed to kind of build up in the atmosphere, right? And roundabout, it's very interesting. So here's this increase in atmospheric oxygen, and it isn't until right about that time, or a little bit after, around 2 billion years ago, that we see the very first occurrences of phosphorites in the rock record. Okay, so all the way back in here, we have, you know, lots of rocks preserved, but we don't have any phosphorites from these times. Right here, we start to see phosphorites showing up on basically all the continents. And here's a map that shows them, in fact. So here, these are all the phosphorites from around that age. And you can see here 2 billion, 2 billion, 2 billion, 2.3, 1.9, 2.1, and so on, right? So here 2.0. Yeah, so all around this time here, we have these phosphorites showing up all over the world. And if we look at those phosphorites, if we cut them open and look inside under the microscope, we can actually see some very interesting features. Here are these little phosphatic rods that all have the kind of similar size, and they look quite a lot like phosphatized bacteria. Ivo Lepland and colleagues suggested that some of these larger granules that are kind of covered or enclosed, lots of these smaller little rod-shaped structures are actually larger, possibly Thiomargarita-like organisms that are preserved. I'm not sure if that's correct or not, but these are very interesting features in these 2 billion-year-old phosphorites. We also see in rocks from India that are 1.7 billion years old, we see these kind of interesting, it's hard to make out with this lighting, but basically these kind of twisted stalks of iron oxides. And these twisted stalks of iron oxides are exactly what are produced by iron oxidizing bacteria today. So here this is an image that shows an iron oxidizing bacterial cell that basically produces these twisted stalks of iron oxides. And you see basically inside of some of these oldest phosphorites, they're actually phosphatized stromatolites. They contain lots and lots of these twisted stalks of these filaments that look very, very much like what's produced by modern iron oxidizing bacteria. And those modern iron oxidizing bacteria, not only do they make polyphosphate today, but they also kind of are indicative of these kind of dynamic conditions with respect to oxygen, right? So you have these gradient conditions that they require, and they're in conditions where oxygen can fluctuate and so on, which is most likely why they also store polyphosphate. Similarly, other deposits in India, we see something that looks quite similar to Beggiatoa, so there's kind of this septate morphology here. Could be cyanobacteria as well, but they're quite large filaments that are septate. And then these kind of spherical features here that are hollow inside, maybe they represent something like Thiomargarita, maybe not. But again, these are all some of the oldest phosphorites, and they contain lots and lots of these microfossils that, at least in terms of their vague morphology, kind of resemble sulfur oxidizing bacteria. And then for another interesting example from the rock record, so there's this big rise in oxygen here that correlates with this first peak here in phosphorites. But then they kind of seem to go away for a while. And then right here, when we see this final kind of uptick in oxygen during the Neoproterozoic times, which are about 600, 700 million years ago, that's when we start to see this other rise, the second rise in oxygen up to levels that are characteristic of the present day atmosphere. And again, that's where we see this next correlation between phosphorite abundance, the appearance of phosphorites on every continent, and oxygen. And so here, actually, this is when phosphorites first appear in kind of mineable form, the really kind of expansive phosphorites on many, many continents. And in those, we also see some very interesting features. So here, I'm in southwestern China, and I'm in a phosphorite mine. And if you zoom in at what I'm pointing at, this whole kind of mountainside that I'm pointing at is made up of lots and lots of these little phosphatic grains. So all these little white specks in here are all phosphatic grains. Most of them are spherical grains. And if you dissolve those out of the rock, you can see basically if you dissolve away the kind of carbonate matrix or the surroundings of them, there are a bunch of these little fossilized spherical structures here. The whole mountainside is basically like that. Billions and billions and trillions of these things. And these are from the Doshantou Formation in China. They're about 600 million years old. And if you look at these things under the scanning electron microscope, there's not just an individual sphere, but there's basically clusters of what appear to be fossilized cells that have these division patterns that are actually very similar to what's found in animal embryos today. So you see these kind of clusters of cells here. And so a number of researchers had put forward this idea that these fossilized cells here, these ancient ones from the Doshantou, that they actually represent the oldest evidence of animal life in the form of fossilized embryos. Right? So these things here, you can see this is actually an SEM image of the Doshantou microfossils, and here, this is all these here are actually showing this blastula, this early stages of a sea urchin, right? It's a sea urchin embryo. And so you can see there's actually quite a strong resemblance. These features are quite large, so they approach millimeter in diameter, so about the same size as modern embryos. And they have this so-called apparent reductive division. So what that means is basically instead of getting larger and dividing, the fertilized egg divides in 2 and then divides into 4 cells and 8 cells and 16 cells and so on without increasing in volume, right? So the number of cells is increasing with each division, but the total volume of the entire mass is not. And so that's actually something else that's observed in these Doshantou fossils, which wouldn't be found in something like algae, for instance. And some of them are surrounded by this kind of envelope. So these lines of evidence were what were used to put forward the idea that these things are animals. There are a couple of structures that have been interpreted as adults, but they're very controversial, and many people don't find them convincing that there are anything even resembling adults in any of this population of these billions and billions of these so-called embryos, possible embryos. And so when I was a graduate student, I was interested in these fossilized structures, and I was also aware of this whole Thiomargarita phosphorite business. And so one of the things that I came across was basically this report that Thiomargarita from the Gulf of Mexico actually undergoes reductive cell division, and it produces basically structures that look a lot like embryos. And so if I compared basically modern Thiomargarita here on the left with the Dochanteau microfossils here on the right. So you can see they look pretty similar, right? Here you can see there's lots of these kind of arguments about the division planes, the way the division, there are certain shapes of the division planes that are similar between the Dochanteau and Thiomargarita and animal embryos, right? So there's now another possible candidate for interpreting these Dochanteau microfossils. They could instead just be Thiomargarita of the kind that actually are making phosphorites, we think are making phosphorites in the modern day. But after that, there came about other reports of fossils in the Dochanteau formation that didn't seem so consistent with Thiomargarita. There are things that out there that have this kind of, these little spiny looking deals, projections coming off of this envelope that's around what appears to be those dividing cells. So this kind of thing is not something that we see in Thiomargarita. Strange features like this, these like track of these little pits. And there are lots of other, there are a number of other features there that seem more like eukaryotic material than they do Thiomargarita or bacteria. But one of the features that they particularly call out as being important is actually this textured envelope. So sometimes these little spheres are covered with this kind of polygonal shape textured envelope on the outside here. So this layer. And they say that that's very characteristic of animal embryos. But in fact, we've actually just produced, using no biology whatsoever, just chemical experiments, precipitation experiments, we can produce this spherical structure with these multiple layers and this polygonal kind of outlying texture here. And we can even get precipitates that have these kind of division planes in them. And these are completely non-biological. They're grown in a test tube with just calcium and phosphate and some gelatin matrix for them to precipitate in. So I'm not saying that these Dochanteau fossils are abiotic precipitates. Many of them have clearly biological features that we can't really produce in the lab, but the point here is that there are lots of features that we can see in microfossils that might actually be the result of non-biological processes. So we have to be really careful about what we use as a feature to say, oh, this is characteristic of some living clade, and so we can use this morphological argument to say, oh, this is definitely animal embryos, or this is definitely sulfur bacteria. It's very complicated, and we have to be very careful. There are, to return to just using morphology for this argument here, in the Dochanteau formation, there are these filamentous features here that have these septate filaments here, these septate filaments that look a lot like Beggiatoa. But here we are able to show that, in fact, in these filaments, they actually contain little sulfur inclusions that are very similar to what we see in Beggiatoa today. So modern Beggiatoa has all these sulfur globules. We see stuff like that actually preserved in the organic material that's associated with these fossils in the Dochanteau. So remember, Thiomargarita and Beggiatoa are like really close relatives, and so they're oftentimes found together or in similar environments. So we think this represents, bona fide evidence of sulfur oxidizing bacteria in these ancient Dochanteau deposits, regardless of whether or not those spherical aggregate forms are actually animal embryos or sulfur bacteria or some other kind of eukaryote, or actually what I think is more likely is there's an assemblage of a bunch of different stuff, and some of it looks superficially similar to one another, and really you're seeing this kind of a deposit that represents a lot of different types of organisms. In the modern, we see actually with Thioploca off of Costa Rica, we can actually see sheaths of Thioploca that are partially phosphatized. So this is actually still biological material, but out here, this is all, these filaments are actually now apatite. So they've been turned into phosphorite. So this seems to be like actively happening. We think we see similar mineralized cells of Thiomargarita off of Namibia. We're still in the process of analyzing these in terms of the chemistry. And so together, the hypothesis here is that basically we see phosphogenesis in the modern associated with this polyphosphate metabolism. And if we go back into the rock record, we see microfossils that at least superficially resemble polyphosphate accumulating bacteria in numerous ancient phosphorates. Phosphorates from all over the world. And during the Precambrian here, so way back in deep time, these phosphogenic episodes, these times when we see phosphorite showing up here and here, they roughly correlate with episodes of oxygenation, right? So this increase in oxygen, and you get your phosphorates. So in modern day, polyphosphate metabolism is important for organisms that have to deal with these kind of complex changing oxygenation conditions or oxic-anoxic gradient conditions. And so hypothesis is, did the kind of initial development of oxic-anoxic transition zone conditions 2 billion years ago, as you started to have this rise in oxygen in the atmosphere, did that kind of lead to favorable habitats for the polyphosphate accumulating bacteria? And then those bacteria, did they then create the conditions that were favorable for those phosphogenic episodes during those times? Right? So that's still a hypothesis, still an open question. I think it's an interesting idea, but there still needs to be a lot more work done. So it's a nice story, but is it too simple? I think it is, and I'll present some evidence from the deep sea just to kind of wrap this up. So this association between Thiomargarita and polyphosphate metabolism and the phosphorite formation, it's only really been studied at a few sites and really only, I think, rigorously shown here off of Namibia. But there are other places that we can study sulfur bacteria. It's not so easy to get up to a lot of these places. It's not so easy to get ship time to a lot of these places and go to cruises and so on. It's one of the reasons I'm so excited to be here, is to actually be in a place to study where sulfur bacteria are found and phosphorites are found. But there was one site that I had an opportunity to go to, which wasn't kind of a canonical phosphorite zone, but it was a place that had sulfur bacteria. And so I had an opportunity to work off of Barbados, where there are these mud volcanoes. So they're kind of like a methane seep. There's fluids coming up from deep, and basically you have all this methane coming up, and it gets oxidized by the anaerobic oxidation of methane by archaea and the consortia of archaea and sulfate reducers. And it produces these sulfitic conditions that then allow these Thiomargarita-like bacteria to grow. And so this is a different organism. It's called Thiopalula. And basically what Thiopalula is it's kind of like a mini Thiomargarita, which sounds weird, but that's what it is. So it looks a lot like Thiomargarita. It's like this spherical cell it's all full of sulfur inclusions here, and it makes these mats. And you can see here, this is the ROV Jason that's coring these mats. And this is at 5,000 meter water depth. So pretty deep for these Thiomargarita or Thiomargarita-like bacteria. And these are not phosphogenic sediments. There are phosphorites near Barbados, but these particular ones, we aren't aware of any phosphorites in this vicinity. But we thought, well, whether we have an opportunity to kind of study to see if we actually see kind of this phosphate release from these sulfur bacteria if we expose them to anoxic conditions. Would we see that? That's what we wanted to see. And so my former postdoc, Dan Jones, he basically did these incubation experiments, and he showed basically a phosphate release that was kind of similar magnitude to what was shown with Thiomargarita. But what we did is, what Dan did is basically he took these different incubation samples and he extracted the RNA from it, and he made what's called a metatranscriptome. And so metatranscriptomics basically looks at, analyzes the messenger RNA in an environmental sample to basically show what genes are being expressed. So if you're looking at a genome, you're just looking at genetic potential, right? But if you're looking at a transcriptome, you're looking at what genes are actually being expressed under those conditions. So what we did is we took the sediment samples and we exposed them to either aerobic conditions or anoxic conditions, and then we looked at what genes were being expressed. And so we thought we'd see genes that were associated with polyphosphate metabolism. What we saw was genes with these Thiopula, these little mini Thiomargarita, that were associated with sulfur oxidation. So this reverse dissimilatory sulfite reductase, that's an important component of one of the pathways that's used for oxidation of sulfur by sulfur bacteria. So that's what we expect to see, something like that. We expect to see transcripts of genes involved in sulfur oxidation. We also saw genes that we think are involved in using nitrate, the reduction of nitrate to ammonia. So we saw like hydroxylamine oxidoreductases and nitrogen transporters and so on. So things that we expect, again, we saw gene expression for what we thought we would expect based on what we know about Thiomargarita's biology and Thiopula's biology. So Thiopula was active, and we thought we'd see polyphosphate going on there because we see the release of phosphate. But in fact, what we saw is lots and lots of transcripts for polyphosphate-related genes, but none of them were from Thiopula. None of them were related to any of the giant sulfur oxidizers. Instead, there was a whole bunch of other type groups of bacteria out there that were instead expressing all of these polyphosphate genes. So these genes here, polyphosphate kinase and exopolyphosphatases, basically were being expressed under these different conditions by different bacteria. So you had epsilons and alpha proteobacteria, Firmicutes, so on. The epsilon proteobacteria are actually sulfur oxidizers, but they're little sulfur oxidizers. But we didn't see anywhere in this actually any sign of Thiopula. That doesn't mean that they weren't metabolizing polyphosphate. We can't really conclude that based on this transcriptome because maybe we just didn't actually identify those genes, or maybe it was just detection limit in terms of the number of cells and so on. But what this does show us is that the story of polyphosphate metabolism in these sediments is much more complicated than we thought, and that there are lots and lots of other organisms out there that could be storing polyphosphate and releasing polyphosphate, not just these giant sulfur oxidizing bacteria. So other bacteria seem to be involved, and our sulfur bacteria that we thought were so important, at least at first look, don't seem to be so important, or at least under those conditions we weren't able to detect those genes. And so I've added 2 question marks here. Initially I had 1. So that's oftentimes what happens in science, right? You go from 1 question mark to 2 or 3. So really what we want to do just going forward is we want to expand our kind of studies of other phosphogenic zones, other zones where we have Thiomargarita, other sediments that don't have Thiomargarita that may be exhibiting kind of similar phosphorus cycling dynamics, even though maybe they don't result in a phosphorite. That would be interesting. Then we might think that something else is going on. Something else in addition to this polyphosphate metabolism might also be important. So we want to look and see, what other organisms are involved in polyphosphate metabolism, under what conditions are they actually taking up the phosphate, under what conditions are they releasing it, are there other mechanisms that are involved in addition to this polyphosphate metabolism that lead to phosphorate formation? We don't know that. It's possible. Maybe there's an accompanying change in pH that's associated in a change from aerobic to anaerobic metabolism that accompanies this phosphate release. So there could be other things going on. Where is it happening? In many cases, the spatial association between Diamargarita, and where we find the phosphorite accumulations are kind of separated. And as we know, when we look in the sediments in many systems, there's lots of heterogeneity, right? So where are these actually occurring, right? And finally, how important are this polyphosphate metabolizing bacteria for phosphorite formation? How much are they really accumulating? Are there other processes that are also important? And so I just want to thank funding agencies and all of the people in my lab, especially Dan Jones, who did a lot of this work. And with that, thank you.
So from past to future, thank you for this promising and inspiring lecture. Time for a question, please.
You have one related to this polyphosphate accumulation is related to these changes in or peaks of oxygen.
Mm-hmm.
And when these peaks occur, there's changes in the biochemistry metals, and iron is lost the solubility and its copper is more available. Maybe one of the interesting questions about how these changes in trace metals chemistry can alter this phosphate accumulation or the expression of this one. And it's interesting because also in the other lectures, some of the remarks is that there's a strong presence of genes involved in copper metabolism in the deep ocean microregions. So maybe there's been some link.
Yeah, that's a very interesting point. Yeah, I think looking at other kind of trace elements over time and how trace elements responded to, say, oxygenation of the oceans and so on, and oxidative weathering of the continents. That's another aspect that I didn't really have time to talk about, but you know, those correlations between phosphorites and oxygen, there's another explanation, or another part of it is that you know, you have the onset of oxidative weathering of the continent. So you have more phosphorus being derived from weathering of rock material. And you might also have more input of other kind of trace elements, trace metals, and so on at those times as well. Maybe some of those could be recorded in those phosphatic mineral deposits. And you could kind of backtrack and see if other trace elements, as you say, might also have been important by seeing if they're kind of preferentially preserved in those deposits.
Any other question?
Osvaldo?
I'm still trying to understand what would be the advantage of storing this polyphosphate.
It's an energy reserve. Yeah, they can basically make it, convert it into ATP. Yeah. So they. It's basically, I mean, the energy is stored in the bonds basically, just as it is with ATP, right? So you can hydrolyze those bonds and couple the energy with a bunch of other biochemical. Yes, yeah. For polyphosphate, yeah. In fact, you know, there's a lot of literature on polyphosphate metabolism broadly as it's related to fundamental kind of biochemistry and metabolism, but in most cases, they're not really looking at this polyphosphate accumulation, right? So oftentimes, like I said, it's just transitory. So it's kind of used as like short-term stress response, energy storage stuff, but then this polyphosphate accumulation, it's only a certain subset of organisms that actually store lots and lots of it. But basically, they get energy. They store energy in it. Yeah, and then they use that energy when there's a stress response or a change in environmental conditions. Yeah.
Is polyphosphate also stored in diatoms, for example?
Yes.
It is like with some staining a long time ago, and I found it in the spines, really.
In the spines, huh?
Yeah.
Huh.
Like in Cathosaurus chains, that they would have it in the spines. I mean, someone told me that these are polyphosphate bodies.
Yep, that's right. Yes, that's true. So there are diatoms that can accumulate polyphosphate, and in fact, The presence of polyphosphate extracellularly or like in phytodetritus, that's also a possible mechanism or substrate by which you could have apatite precipitating. So basically when you break down that polyphosphate, heterotrophs are breaking down the polyphosphate, you have the release of phosphate and you'll have the release of calcium because in many cases, particularly with eukaryotes, they store calcium along with the phosphate in the actual inclusions of polyphosphate. They call, like these, in eukaryotes, it's often called acidic chalcosomes because they have this, they're acidic and they contain phosphate and calcium, but they're basically polyphosphate granules. And so, yeah, that's another possible mechanism, like even off of Namibia, yeah, the diatoms could be a contributor to that as well, for sure. Doshanto.
If those are archaea that are there, especially if they're methane oxidizing archaea, could you be looking at the isotopic characteristics of the material there, or could there be some long-lived organics like isoprene units that would give you an idea of whether it's eukarya or archaea?
That's a good question. So in the Doshanto Formation proper, in those rocks, the organic material has been thermally altered to too much for the preservation of lipid biomarkers, but you certainly could do that. You know, people have looked in the Doshanto a lot for biomarkers, and they're basically not preserved well enough. Those rocks just got too hot. Yeah, but it's an interesting idea, and certainly something you could look for. And there is a methane oxidation signature broadly in some of the carbonate rocks that are associated with the Doshanto Formation, but many meters up section. And so that's actually in the carbonate rock, it's not in the organic material. So it has been suggested that there was methane oxidation going on broadly in the Doshanto depositional environment, but we can't say that that was specifically associated with the environment of where these fossils were deposited, or, yeah.
Maybe you say that I missed it, but so eukaryotes and bacteria can accumulate this polyphosphate.
I don't think there are any polyphosphate accumulating archaea to my knowledge. I'm sure I'm wrong, but but yeah, I don't know why they wouldn't, you know, but yeah, it's a good.
But there is no report of that.
To the best of my knowledge.
Okay.
Is there an explanation because giant bacteria have this polyphosphate? I mean, a correlation between size and the presence of these polyphosphates? Nothing.
No, we don't think so. We think that this polyphosphate accumulation is more important and more broadly distributed than kind of the literature would suggest. And in fact, you know, you just have to dig into different parts of the literature as well. That, you know, for example, so we're actually working on dental biofilms now and looking. I decided it might be interesting to look to see if there's polyphosphate accumulating bacteria in dental biofilms. Sure enough, they're totally, yeah, so these dental biofilms are full of polyphosphate metabolizing bacteria that could have important implications for basically the saturation condition of your teeth, right? Because if you have too low phosphate in there, then the mineral will start dissolving, right? So most dental researchers are just looking at like acid production. They're not looking at kind of the dynamic sequestration of phosphorus by bacteria that are living on the tooth surface. But they're basically, you know, our teeth are rocks covered with bacteria, right? And bacteria that accumulate polyphosphate. So, you know, even like dental calculus is not well understood, right? So maybe polyphosphate accumulating bacteria are important for dental calculus. So that's what we're looking at now, and we're really surprised about how many different dental bugs actually accumulate polyphosphate, and they're totally different. Their responses are totally different from the environmental bugs. A lot of them are like gram positives, and they behave completely different. They're yeah, we don't understand them, or we're just,
Nothing to do with the size. It's
no,
just
I,
a curiosity with it.
right, yeah, yeah, I don't think it does, yeah.
What about the distribution of the polyphosphate deposit inside of cells? They are distributed randomly or in certain parts? I mean, in the endings, in the center?
I'm trying to think of an example where they're not kind of randomly. I mean, I think of them as being random, more or less. Yeah, I'm not aware of any kind of organization of polyphosphate in bacteria where they occur in the cell. But I'll have to think about that. Okay.
In which moment the bacteria use this deposit? Because it can be used as energy, right?
Right.
So in which moment they can use this deposit?
Most often, at least in the kind of canonical study of, like the wastewater treatment bacteria and so on, it's the transition from anoxic to oxic conditions, or oxic to anoxic rather. Yeah. So they take it up under the oxygenated condition and then they switch back to anoxic conditions. That's when they use it. But we actually have a cultivated Gamma proteobacteria, a small bug, that's related to Thiomargarita actually, but it's not a giant sulfur oxidizer, and it does the opposite. So yeah, when we think we have something figured out, or it turns out, of course, we can find the opposite example, and so it's just, the biological world is complicated.
You showed an image of Thiomargarita that was like racing polyphosphates, maybe?
That was releasing or open to the environment vacuums of sulfur and polyphosphate. What happens to the surroundings of the microorganisms once they die or release these accumulations to what is surrounding them? Like, is a source of polyphosphates to other members of the microbial community?
That's a great question. I imagine so. I imagine that's why you have like polyphosphate hydrolases broadly, is because organisms like diatoms are breaking down in the sediments, and organisms need some way of scavenging that phosphorus, and so yeah, they probably, yeah, they have enzymes to break those polyphosphate bonds, and that could presumably be done extracellularly to get the phosphorus.
So the pollution is kind of stable to be released. It doesn't just degrade into the environment.
If you kill a polyphosphate accumulating bacteria, if you wipe it out and then, so there's been some studies of, Julia Diaz actually did this. She looked at like the polyphosphate and diatoms from a different environment, but she actually did show that there is, they are stable on the seafloor, but then they actually can catalyze this transformation, abiotic transformation, to very small inclusions of apatite material as well. So, yeah. Kurt.
Considering how difficult it is to make a medium without phosphate, have you ever been able to stress your Thiomargarita to use the intracellular polyphosphate rather than phosphate given in a medium?
Oh, well, we can't get them to grow at all. But yeah, you can expose them to anoxia or sulfide, and they'll they'll break down the polyphosphate and release it. Yeah, yeah, they do. That's right. Actually, not with oxygen, with sulfide. Mm-hmm. Yes. Yes.
They wouldn't even need it to grow.
Right. I mean, in these sediments where you have this occurring, I mean, in the water column you might have a couple micromolar phosphate, in some of these sediments, you're talking about several hundred micromolar to even almost millimolar concentrations of phosphate, which is huge. I mean, yeah. So they have a lot of phosphorus available because they're living in diatomaceous oozes and so on. But they also seem to do this at like methane seeps and so on, conditions that are more oligotrophic. So, yeah, it's interesting. I mean, we need to be able to grow them in culture, but all of our attempts so far have failed. I mean, it's like worse than watching grass grow. You can have cells that are clearly like in the process of dividing when you collect them, and then you try to make them happy, and they just sit there and laugh at you. Yeah. So, all right. Well, thanks.